All right, everybody, we're going to be talking about those microscopes I handed you out on Friday, or you're going to get on Wednesday, depending on whether or not you showed up. Either way, let's talk about some microscope safety. The first thing that you need to know, and I kind of already told you about this, is that you need to have both hands on it when you're carrying it. So you need to have one hand on the arm, one hand underneath the base. Okay. Let's see. The other thing that you need to know is that when you are trying to focus your microscope, sorry, this is very clumsy. You have this knob here, okay, and then you have a larger knob. So the larger knob is going to move the scope, or the stage rather, a lot further. The smaller inner knob is going to move the stage a lot less. It's going to be a lot more subtle. Okay, and so it asks you in the lab to write down how do you focus a microscope. You always start on low power. So let's get our light on. You always start on low power. That's the smallest objective. What does this one say here? This one says 4 on it. Okay, so this is my small one. And when you're on low power, then you can take a look in the microscope and you can use the coarse adjustment knob. Okay, you can use the coarse adjustment knob. You can move this thing up and down as much as you want because it's not going to run into anything, even if you get it all the way to the top. Okay, but you get it into focus on low power first. If you don't start at low power, it's going to be very difficult for you to do much of anything. So get it into focus on low power. Okay, we'll pretend like I got mine into focus and I dialed it in. Then you move up to the next highest power, which is this one. It says 20 on it. I believe your guys' scopes are the same. When you're on higher power, when you're on anything beyond low power, you only use the fine adjustment. You only use that small knob. Okay, if you use the larger knob, odds are pretty good that you're going to mess up the lenses by running them directly into the slides, or you're going to break a slide. It's just going to be bad. Okay, so if you're at anything other than low power, only use the fine adjustment. So sort of part and parcel with that, you guys are going to be expected to know the basic parts of your microscope. Okay, coarse adjustment, fine adjustment, iris diaphragm, mechanical stage, oculars, just the basic parts. Just so you guys know, that is going to be something that I expect of you. So now that we've talked about kind of how to focus it and how to use it just a little bit, there's some questions on the lab about magnification. Okay, so your lenses, your oculars, these removable pieces that you look into, these are called oculars. They should be 10x, and it will say right on them what they actually are. Mine says 10x right there. Okay, so if I was just looking through those, it would be 10 times closer. Everything would appear 10 times closer than it actually is. Your objective lenses here, okay, those are the ones that are on that rotating nose piece, the ones that rotate. Okay, those have a magnification written on them as well. Okay, and what you're going to do is you're going to multiply that magnification by the magnification of the ocular. So low power says 4 on it, right? What is that low power times the power of the objective lens, or the ocular lens, I should say? It's 4 times 10, which gives you 40x. It should be pretty easy math. It's just multiplying stuff by 10. So I'm going to let you guys, you have a 20 on here too. 20 times 10 is 200x. You have a 40 on there as well. 40 times 10, 400x. Okay. So one of the first things we're going to be doing in the lab is we are going to be observing a human hair. Okay, and what you're going to do for that is grab out of your bag of goodies, out of this thing, grab out the Petri dish. It's a circular dish. You should see some blue tape in here. And you're just going to grab that blue tape and you are going to pull out your slides. You maybe saw I almost just dropped the stupid thing, so be very careful. You're going to pull out your slides. These are not disposable. I'm asking you guys to try to use these as many times as you can, and it shouldn't be a problem, all right? The one problem you might run into is in this hand there are 2 slides, in this hand there is 1 slide. So you need to be very, very careful and make sure that you're only getting 1 slide at a time. If you have 2, it's going to be too thick to look at. It's going to be too thick to look at anything up close on the microscope. So go ahead. We've got our slide here, and we're going to be observing a piece of human hair. So you're going to need a pipette. So things that you need so far: a microscope slide, a pipette, a small amount of water, and either a hairbrush or just your head. You can also look at other kinds of hair. You could look at like your arm hair. You could look at the hair from your pets if you want, but you need to at least be looking at 1 strand of human hair. Okay, so I'm just going to go ahead and get a little tiny bit of water. I'm just going to put 1 drop. That's all you need, is a single drop here. I'm going to go back to my brush. And I'm going to grab 1 single strand of hair. And what you're going to want to do is just kind of drag it through the water so that it stays flush. You probably can't see that, but I just put a drop of water on my slide and then I just drug the hair through it so that it sticks to the slide. You don't actually need a cover slip to look at hair. But yeah, again, the proper focusing of the microscope: start at the lowest power that you can. When you're on low power, use the coarse and fine adjustment knob to get your specimen into sharp focus. And then, only then, do you move up to the next power level. Okay? The other important thing, and that must be written down under proper focusing of a microscope, is that you only use coarse adjustment on low power. Okay? And the other thing is, these scopes they're referred to as parfocal. That's not a word that you need to know, but it means that in order to be able to see much at medium power, you kind of have to have it in focus at low power first, because once you move up, then your object will still be in focus. The whole point of me saying this is just that you have to start from the bottom. You can't just start at high power and be there. Okay, so we've got our human hair slide all done. Let's see what's next. Proper focusing of the microscope. We already talked about some of these things. So estimation of field of view. I'm going to go ahead and stick a ruler up on canvas that you can print out, and you can use that to help you estimate the field of view. Okay. Because I'm going to ask you to be estimating a few different things. Like I'm going to ask you to estimate the width of a human hair just by looking at it. Okay, cheek cells. Pause this and get a toothpick. I just realized we aren't doing cheek cells till last, but that's okay. Basically what you're going to do is you're going to take your toothpick and you're just going to rub it along the inside of your cheek. You don't need to stab yourself or anything, but press a little hard, maybe on your gums, see if you can get something. Because you need a little bit of a sample here in order for this to work. All right, so I got my little cheek sample. I'm going to smear it here. You're really going to want to be able to see your cells on here, okay? You can also get some, if you have some up by your teeth, that's a good spot to get some cells too. Clean teeth today, I guess. Okay. Yeah, and you're gonna be able to see it on the slide. You won't be able to see it on here, but you should be able to see that you actually have something there on your slide. Okay. So what do these instructions say? Get a dropper and put about 2 drops of water onto the specimen. Oh, no, I'm sorry. I was reading the wrong thing. Smear the end of the toothpick on the glass slide, throw the toothpick away, let dry for a couple of minutes. Then you're going to, the next thing that you're going to need is out of your bag, inside of this bag, there is a small dropper bottle that looks like this. Mine says meth blue, yours will probably say methylene blue. Okay, what does it say to do with the methylene blue? 2 drops of methylene blue over your specimen after we've let it dry out. The lid didn't want to come off. So, yeah, 1.5 drops is probably fine, if that makes any sense. You don't need huge drops for this to work out. So 1, 2. Okay, make sure it covers up your entire specimen. And that says use the water dropper to put 2 to 3 drops of water on the edge of your specimen. Okay, so we'll do that. And again, that doesn't, you don't need a ton of liquid here. Get myself back to full screen. Okay, so there's my sample with my methylene blue. 1, a couple drops. Okay, so this is way too wet, by the way. That's not the end of the world. But there's too much liquid on my slide right now. And so actually what I'm going to do is I'm going to grab a paper towel from my kitchen, and I'm just going to dab a little bit off of here. Okay. Slide, we can just dab some of this off. I'm staying away from the cells. This probably not even a necessary step, depending on how good you are. Okay, so I've got my dried and dyed slide here. The next step that you need to do is you need to get out a cover slip. This should also be inside of your Petri dish, which we're going to use for an experiment later on. But be very, very careful. These are super, super thin. You can easily break these in your fingers. Okay, so just want to very, very, very gently, very carefully pull out a cover slip. I'm going to try to have you guys reuse these too. It might not work out. And as I said in the lab, if they break, really not the end of the world. All right, at long last. We have our sample here. I have my cover slip here. I'm not sure how well you can see this, but I'm holding it at a 45-degree angle, and I'm just going to let it drop. Okay, and that should push all the air out of the way and just leave you with a nice, I dropped my cover slip because I was playing with it. But it should just leave you with a nicely prepared sample of your cheek cells. What's next? And be careful with the methylene blue. This is a stain. It will ruin your clothes. Okay, so don't wear anything that you particularly care about while you're trying to run these labs. Got some questions about Elodia. Let's go back up. Okay, obtain a slide and 1 leaf of Elodia. So Elodia is another thing, another one of the goodies inside your bag. I left mine out somewhere. My Elodia, and you're just going to pull off 1 leaf. Mine is definitely looking worse for wear, so get to this early in the week if that's at all possible. Okay, so you've got our Elodia here. I'm just going to try to get 1 leaf onto that. Okay, there is my Elodia leaf on my glass slide. Let's see what the other instructions are here. Use the water dropper, 2 to 3 drops on the specimen. I think that's excessive. Make sure you have a clean water dropper. I think you probably could get away with just 1. I'm putting 2. Just want to make sure that it's completely covered up. Okay, and once we have that, we're going to use the same process that we did before with our cheek cells. So we have our wet slide. You're just going to take your cover slip, hold it at a 45-degree angle or so, and just drop it. Okay, and so there is your prepared Elodia slide. I'm going to have you guys go ahead and draw that for me. The other thing that I'd like you to do, I don't have it written in here yet, but I would like you to take your, another tube in here, maybe it's in your chemical bag, but you need to find your NaCl solution. And I want you to do the exact same preparation that I just did, but instead of using the water droplet from here, I want you to use that NaCl solution, okay? And, you know, make sure you're using like a different pipetter every time, just so that you're not getting like methylene blue inside your salt water, or you're not getting salt water inside your fresh water, although it's less important. You can just pour that out. Okay. So what else do we have to talk about? We've got our Elodia slide ready to go. We've got our human hair and cheek slide ready to go. We have some questions about these cells. I thought there was one more. Onion. Yes, onion. So if you guys have an onion, that's great. If you got to go buy one, please, you know, try to just go buy one or borrow one from a friend or something who's not going to get you sick. You're going to do the same. If you can see, it's just peeling. This is the layer that you want. Very thin. Okay, you just want to use your fingernail and scrape this outer skin off. Then you're going to put this onto a slide flat. Couple of drops of water and a drop of iodine. And the iodine is going to stain that onion, because these onion cells are clear. The ones that we're using, they don't have any pigmentation to them. And so we're going to stain that with iodine. Okay. The reason we put methylene blue on your cheek cell is because your cheek cell is also clear. If it was not stained, you could maybe see a couple of things, but you wouldn't be able to see it very well. Okay, so I think that is pretty much it. You're going to be answering some questions down here. I'm going to have you be estimating how large things are using your knowledge of each of these. So again, this is a micrometer unit that you'll learn in the scientific method lab. But if your magnification is 40x, your field of view is about 4,500 micrometers. And so if a cell in there, or if some organism in there is taking up half of that amount, how large should that be? Right? It should be about half of 4,500 micrometers. Okay, I've already covered this stuff. Yeah, make sure you fill this in before you even get started with today's lab. And I'll be online for office hours this coming week. Please let me know if you guys have questions about anything. All right, very good. See you soon.